Shown here is a neuron. A neuron is an electrically excited cell that transmits and processes information. The diagram shown here is the anatomy of a neuron. First things first is the cell body, which contains all the organelles of a neuron. The black dot here represents the nucleus. A neuron has several branches. The fine branches and several branches are the dendrites, which receive signals from other cells. Then there is one specific branch, a longer branch, called the axon, which transmits a signal. And where the axon widens to connect to the cell body is called the axon hillock, which generates a signal. The part where the neuron connects to another neuron is called a synapse, which is the junction that transmits the signal across to another neuron. There are synaptic terminals at the synapse, which are the ends of one neuron. The green arrows shown in this diagram represent the pathway of a signal. The signal is received from another cell through the dendrites, as mentioned before, then travels down the axon and through the synapse, which is the junction, to go through another neuron and jumps from neuron to neuron. Depicted here is a more detailed diagram of the process that shows the transfer of a signal from one neuron to another. The first neuron contains a presynaptic membrane, and this contains a voltage-gated calcium channel, which is activated by the action potential. This action potential, therefore, causes an influx of calcium into the presynaptic membrane. This influx of calcium also promotes the fusion of a vesicle, the orange circles, and which contain the red little circles, which represent neurotransmitters. The fusion of the vesicle with the membrane causes the neurotransmitters to escape into the synaptic cleft, which is the space between the two neurons. And therefore, the neurotransmitters diffuse across the ligand-gated ion channels into the postsynaptic membrane of the other neuron. If you direct your attention to the upper right corner, you will see a more magnified view of the relationship between a neurotransmitter and the ligand-gated ion channels. The neurotransmitter binds to the receptor part of the channel, and in this channel, both sodium and potassium ions can diffuse through the channels. When the neurotransmitter is released from the receptors, the channels close, and so both sodium and potassium cannot travel through. Synaptic transmission ends when the neurotransmitter diffuses out of the synaptic cleft. It is taken up by the synaptic terminal or by another cell. In this diagram, we see the plasma membrane. The plasma membrane consists of many parts which involve the passage of potassium and sodium ions. Potassium is the dark orange squares, and the channels that allow them to pass is the dark purple channels. As you can see, there are arrows where the orange squares pass through. There is also a blue channel where the pink circles pass through. The pink circles represent sodium, and so the blue channel is the sodium channel. To the far left, you see a green channel. This is the sodium-potassium pump because it allows the passage of both sodium and potassium. In the outside of the membrane, you see more of the sodium ions. Therefore, it is positive, as you can see as the signs indicated above the bilayer. Below the bilayer, you see a negative sign. That is because there are less sodium and more potassium. There is a net movement of positive or negative charge, and this will generate a voltage or potential across the membrane. This movement is caused by the ions diffusing through the membrane, and each channel is selectively permeable. Therefore, this allows the creation of action potential or resting potential or more. The resting potential is between -60 and -80 millivolts.
The rest period is known as the resting potential in the action potential graph. The resting potential is the electrical potential of a neuron or other excitable cell relative to its surroundings when not stimulated or involved in passage of an impulse. A stimulus starts the depolarization of the membrane. Depolarization is caused when positively charged sodium ions rush into a nerve cell. As these positive ions rush in, the membrane of the stimulated cell reverses its polarity so that the outside of the membrane is negative relative to the inside. Once the electric gradient has reached the threshold of excitement, the downswing of repolarization begins. The channel that let the positive sodium ion channels through close up, while channels that allow positive potassium ions open, resulting in the release of positively charged potassium ions from the neuron. This expulsion acts to restore the localized negative membrane potential of the cell, bringing it back to its normal voltage. The refractory phase takes place over a short period of time after the depolarization stage. Shortly after the sodium gates open, they close and go into an inactive conformation. The sodium gates cannot be opened again until the membrane is repolarized to its normal resting potential. The sodium-potassium pump returns sodium ions to the outside and the potassium ions to the inside. During the refractory phase, this particular area of nerve cell membrane cannot be depolarized. Therefore, the neuron cannot reach the action potential during this rest period. Electrical signals travel faster in axons that are insulated with myelin. Myelin, produced by glial support cells, wrap around axons and help electrical current flow down the axon. Myelin insulation does not cover the entire axon. Rather, there are breaks in the wrapping. These breaks are called nodes of Ranvier. The distance between these nodes is between 0.2 and 2 millimeters. Action potentials traveling down the axon jump from node to node. This is called saltatory conduction, which means to leap. Saltatory conduction is a faster way to travel down an axon than traveling in an axon without myelin. Chemicals, called neurotransmitters, are released from one neuron at the presynaptic nerve terminal. Neurotransmitters then cross the synapse, where they may be accepted by the next neuron at a specialized site called a receptor. The action that follows activation of a receptor site may be either depolarization, an excitatory postsynaptic potential, or hyperpolarization, an inhibitory postsynaptic potential. A depolarization makes it more likely that an action potential will fire. A hyperpolarization makes it less likely that an action potential will fire. Excitatory neurotransmitters are chemicals that increase the chances that a nerve cell will transmit a signal. GABA is made in brain cells from glutamate and functions as an inhibitory neurotransmitter, meaning that it blocks nerve impulses. In the brain, dopamine functions as a neurotransmitter, a chemical released by neurons to send signals to other cells. This is an excitatory neurotransmitter. The brain includes several distinct dopamine pathways, one of which plays a major role in reward-motivated behavior.
Undershoot/hyperpolarization is when the potassium channels open so that potassium ions can leave the neuron. The sodium channels, however, remain closed. As this happens, the membrane reaches its resting state. A synaptic vesicle is basically a membrane-bound compartment that is used to hold neurotransmitters. When there is an influx of calcium, the synaptic vesicle fuses with the presynaptic membrane and releases the neurotransmitters. The postsynaptic membrane is the plasma membrane of the neuron that receives the signal. So when neurotransmitters are released by one neuron, they go and attach to the postsynaptic membrane of another neuron.